Hello, good morning, student. How are you? You are. You all may be as a fine. Now, what are the point we are discussing? So, we learning the chapter number first. The name of our chapter first one is a strength of organic acids and bases. So far we have the second last point remaining in this chapter is application number 6. We are talking about the heterocyclic basis, right? The bases, there are 3 bases, we know that. One is a aliphatic basis, aromatic basis, and the last one is a heterocyclic basis. Aliphatic basis, there we have seen 4 to 5 applications. Now here also we have in a aromatic heterocyclic basis, there also we have seen 5 application. Now is a 6 last one is a application number 6, which is very important as far as the exam point of view is concerned, or this question may appear in 2 different ways. One may be as a like this 2 4 2 4 6 trinitroaniline is 40,000 times 40 times weaker base than 2,4,2,4,6 trinitro n dimethyl aniline. Explain. Or this question may be appear in another way. What is steric effect? What is steric effect? Explain it with suitable example. What is steric effect? Explain it with suitable example. Suitable example. Now in 2 ways, but answer is in the same way. So due to the steric effect, why it becomes as this one 40,000 times weaker base than the second one. So first, what is the first thing required while answering the organic questions? So you must know the structure of the organic compound, then and then you can able to answer this type of the questions. Now this is the compound 2,4,6 trinitroaniline. I think everyone knows the structure of aniline. So let us look at here. The structure of aniline is this one. This is a benzene ring, and one of the hydrogen atom get replaced by NH2 group, and therefore this is the structure of aniline. Now at 2 position, I am, for sake of convenience, I am writing this structure for this way, and here is at nitro N O minus, then double bond O, positive charge on this nitrogen atom, then N O minus, double bond O, again positive charge I am placing on nitrogen atom, and then NO2 group which is present at this position. Now why 2,4,6 trinitroaniline? This is the IUPAC name of this compound. This is the main functional group they have considered here. So numbering start from here 1, either clockwise manner or anti-clockwise manner, you can give the number 1, 2, 3, 4, 5 and 6. Look at here at 2 position, 4 position and the 6 position. There are nitro group, 3 nitro groups are there, and therefore it is called as a 2,4,6 trinitroaniline. Now look at here the hydrogen atom which is electropositive in nature, and this oxygen atom is electronegative in nature, and they get associated via intramolecular hydrogen bonding. This intramolecular hydrogen bonding or hydrogen bonding are shown by dotted line like this, and here also as a intramolecular hydrogen bonding present over there in this compound. This is a 2,4,6 trinitroaniline. This is the structure of 2,4,6 trinitroaniline. Now let us look at the structure of 2,4,6 trinitro NN dimethyl-aniline. What will be the structure of that? The structure of this here, this is a structure of benzene. Now one of the hydrogen atom NN dimethyl-aniline. NN dimethyl-aniline, we know that this is a nitrogen atom, and both the hydrogen of that nitrogen get replaced by methyl group, and therefore this is the structure of NN dimethyl-aniline. And now other 3 groups which are present at this position, so you can write as here NO minus double bond O, positive charge over there, NO2 group, and then again is a NO minus and then double bond O, positive charge over there, and this is the structure of 2,4,6 trinitroaniline, trinitro NN dimethyl cyclohexane. So this is the structure of 2,4,6 trinitro NN dimethyl-aniline. Again the nitrogen atom, and in general those organic compound having as a nitrogen atom are basic in nature, because nitrogen has a lone pair and it can donate that lone pair, and that's why they behaving like as a basis, Lewis basis. And now look at here, now listen carefully. What is the steric effect? Steric effect, look at here at ortho position, this nitro group and this methyl group, they are the bulkier group, this bulkier group, and they create lot of steric interactions are there in this case. But as compared to that methyl group, this hydrogen atom is a smaller, and they get associated with that oxygen atom, and that's why this molecule becomes as a stable one, and this lone pair and the benzene ring, carbon-carbon double bond, they are becomes as a parallel here. Here becomes as a lone pair is a parallel, and therefore they can it involve in a delocalization, and therefore this is this lone pair which is not available for the donation as compared to that one and that lone pair, and so that after there are 3 nitro group, they are also involved in the delocalization. It takes as a longer time, and therefore it is a weaker base. Whereas in this case, what is happening there, methyl group and that nitro group, they are the bulkier group. There are a lot of steric interactions are there, and when trying to push to each other, and they try to stay there, and due to this reason the bond between carbon and nitrogen get rotated, and when the carbon and nitrogen bond get rotated due to the steric interaction, what is happening, the lone pair and that benzene ring, that lone pair and benzene ring becomes as a perpendicular, and therefore delocalization is inhibited here, prohibited here, and therefore it is readily available for the donation, and that's why this compound is a 40,000 times stronger base as compared to this one. This is a weaker base, and this one is a stronger base. And what is steric effect? This is also as a same explanation of this. What is steric effect? And due to that steric effect, this compound becomes as a 40,000 times stronger base as compared to this one. So this is a explanation. So in this way we have completed as a application of aromatic bases. Now let us move toward the third one, which is a heterocyclic basis. As we know that heterocyclic basis means what? One of the carbon atom get replaced by the hetero atom, mainly as here is a nitrogen atom, and therefore they are the basic in nature. So here we are going to see only hardly two examples, two applications. This is a third one is a heterocyclic basis, and we know that heterocyclic basis means what? Heteroatoms are there, cycle is there. For example, first application, let us see here. First application, pyridine is weaker base. Pyridine is weaker base than piperidine. Pyridine explain. This is as usual. For what is our first job, first business, to draw the structure of organic compounds, and as far as my knowledge is concerned, everybody knows the structure of pyridine and the piperidine. Now let us look at the structure of pyridine. Is this one. So simple structure, benzene ring. One of the hydrogen atom get replaced by the nitrogen, and this is the structure of pyridine, and it is a weaker. And now piperidine, structure of piperidine is this one. Structure of piperidine, this one. This is the structure of piperidine. Now we are comparing. This is a weaker base. Why it is a weaker base? Look at here. This is aromatic compound. The lone pair is not involved in the delocalization, but it is readily available for donation. Therefore, it shows as a basic in nature. Now, but this one is a weaker base. Now this one is a stronger base. Why? Because there are this carbon atom has a sp3 hybridization, and as we know that sp3 hybridization shows or exerts plus I inductive effect, and as we know that plus I inductive effect increases the basicity, whereas you look at this carbon atom shows as a sp2 hybridization, and that exerts as a minus I effect, and minus I effect decreases the basicity. Hence the pyridine is a weaker base than the piperidine. So this is the answer of this question. Now let us look at as a second application. Second application is this one, that is a pyrrole. Pyrrole is a weaker base than the pyrrolidine. Pyrrolidine explain. Pyrrole is a weaker base as compared to the pyridine. Again, so simple, that is a heteroatom. Pyrrole is again as a heteroatom. Let us see the structure of pyrrole. That is a 5-membered ring. This is a structure of pyrrole, and the structure of pyrrolidine, it is a saturated organic base. Now again same, whatever explanation we have seen in the previous application, there is a 6-membered ring, this one is a 5-membered ring. That is only difference between these two, and the explanation is the same. But here in this case, the lone pair which is present on the nitrogen, this lone pair is involved in the delocalization, it is not available for the donation, as well as this carbon atom exerts is minus I effect due to the sp2 hybridization, whereas in this case this is a highly basic in nature, because this carbon atom again showing sp3 hybridization and exerting as a minus I effect, and therefore it is a more basic in nature as compared to as a Pyra. In this way we have covered all the application of the organic bases here. Now the last point remaining in this chapter is a catalysis, that is a catalysis, acid base catalysis. The last point remaining in this chapter is a acid base catalysis, acid and base catalysis. Now everybody knows that what is the role of catalysis, and here let me tell you the role of catalyst. It is speed of the reaction, rate of reaction is increased in the presence of the catalyst, and some of the reaction does not occur without the catalyst. If you add as a catalyst, it does not take part in the reaction, but it speed up the reaction, or in a other way we can say that the kinetic barrier is decreased, and therefore that type of the thing is called as a catalysis. And what do you mean by the acid and base? Catalytic amount of acid and base is required to speed up the reaction, and this acid base catalysis is mainly divided into as a two types. One is called as a first one specific acid catalysis, specific acid catalysis, and second one is known as a general acid catalysis, general acid catalysis, and similarly base. Similarly, A is here, that is specific base catalysis, and second one that is a general base catalysis, And now what do you mean by specific acid catalysis? So specific acid catalysis, so specific acid catalysis, it involved in the acidic medium. The rate of reaction is proportional to the concentration of the H plus ion, or we can call it as a H2O, protonium ion. In the water reactions are carried out, and therefore it is directly proportional to the concentration of the H plus ion, and normally these reactions are the rate of reaction normally found to be rate is equal to minus d upon r dash upon dt. As the time changes, the rate goes on decreasing, so that is the general. This kind of the reaction is called as a specific acid catalysis. Now, what do you mean by the general acid catalysis? It also involves the H plus ion, and therefore the rate of reaction is again directly proportional to the concentration of the hydronium ion. Again, the rate of reaction again is a similar one. Rate is equal to minus d upon r dash upon dt, where here is you can take a K1, then R1, R2, like this and so on. So that is called as a general acid catalyst. Similarly, specific base what is specific base catalysis? And this specific base catalysis, it is again directly proportional to the concentration of the hydroxyl ion O minus, or it is directly proportional to the pH. When the pH is increases, the rate of reaction also goes on increasing. So that type of the thing is called as a specific base catalysis. Again, rate of reaction remains as the same. rate is equal to, as a time goes, the rate of reaction goes on decreasing, and therefore it is found to be minus d upon r dash upon dt. Similarly, and the general base catalysis here, other than hydroxyl group also involved there, so that type of the thing is called as a general base catalysis. For example, and the rate of reaction again remains as the same throughout the fourth. Rate is equal to minus d upon r dash upon dt is equal to maybe as a r1 k1 r1 into plus r2 plus dash dash dash. So these are the, I think with this we have completed as a chapter number first. What are the important point you should remember while studying this chapter is a very the weightage of this chapter normally is a 7 to 8 mark appear in the exam. So here 1 mark question may be like this: what is define the acid or define the base according to as a Lewis acid, or maybe as a Arrhenius theory, or what we can call it as a what is specific acid catalysis, what is specific base catalysis, or 2 mark question maybe appear in a question number 3 and question number 4, where the 2 mark weightage of questions are 2 or 3 mark. What is steric effect? Explain with suitable example. Or aniline is a weaker base as compared to the cyclohexylamine. Or trichloroacetic acid is stronger than the acetic acid. Or formic acid is stronger than the acetic acid. Or maybe as a secondary amine is the strongest among the primary and secondary amine. Explain. So in this way you have to study, and if you I think I have taken all this point to point. So I think with this let us stop today here. Tomorrow we will start the next chapter is a second chapter. The name of that second chapter is a disubstituted stereochemistry of disubstituted cyclohexane. Let us stop today here. Thank you. Have a nice day.